Hello, everyone. So, I'll be doing a little presentation on the research that I've done for the past 3 years here at MSU. Even though this is the title slide, I don't know how well you can see it, but I have a really cool picture on here, which I will explain a lot more later on in the presentation, I promise. But basically, so synthetic cardiac development is the broad area. So, we look at cardiovascular health and which I will explore more later. You can see these are actually mini models of the human heart that we've built from scratch with stem cells that you can see here in the image. I did a black background, so hopefully you guys can see them a little bit better. So before I talk about my research, I was talking about me. So again, my name's Haley. I'm a senior here at Michigan State. I'm double majoring in neuroscience and human biology, and I also added a minor in pharmacology and toxicology because why not? I've been doing research since starting my sophomore year of school, so be 3 years by the end of this. That's a picture of my dog. Her name is Stella. I love my dog. That's probably my proudest moment of college. And then the bottom two pictures are just that pipette picture I didn't take. I wish I had those skills, but I work with pipettes a lot. But that is a picture of some of the computer work that I do in lab. But when I'm not doing research or school, I love to read. I read all the time. I just read a book that made me cry so much. I have not cried that much since I had to figure out my physics homework sophomore year. It was super good. So I'm originally from Kalamazoo, Michigan, which is on the west side of the state. Don't know if you guys have been there. It's pretty small, but it's cool. I knew I wanted to leave it, though, for college. So MSU is obviously a great school, as many of you know, and I am really fortunate to have the Kalamazoo Promise, which is basically since I graduated from Kalamazoo Public Schools, I could go to any college in the state of Michigan, and my tuition would be completely paid for, my tuition. So I remember touring a bunch of different schools, and they were nice, but I remember the main reason I was on MSU's campus and I was touring, and I saw in North that they had heated sidewalks. And I was really excited because I'm clumsy. I slip on ice all the time, and I saw that, and I remember I looked at my mom, and I was like, Mom, it's like, this is it. I'm not going to slip and fall here. Just kidding. That was not the only reason, but definitely the first one. And then I knew that they had great academics here. I knew a lot of people. I'm very fortunate that I grew up around a lot of people in the science community. And I had some neighbors that had graduated from state, and they always were, always stating the state gear growing up. And they were telling me how great of a school it was. And I knew that it was a super, super good school for research. And I didn't know exactly what my life plan was going to be going to college, but I knew I wanted to do research. And so on top of the heated sidewalks, there was also phenomenal research opportunities. So now into the fun part. So what do I do? So I work in Dr. Aguirre's lab in the IQ building, which is on the very far part of campus, kind of near the nursing building, if any of you know where that is. So I've been working by the time I graduate, 3 years. I work along my side, my mentor, Sasha, so I'll talk about her a lot. She is super cool, and she does her PhD. And here is basically, if you remember the image from the title slide that I showed you, these are actually how big they are in real life. If you look inside each of the wells, the tiny dots on the bottom of them, those are mini functioning models of the human heart. And so if you would put those underneath a really fancy microscope, it actually has the 4 chambers and beats like a human heart, which is super fascinating, really, really cool. And I have some more images further in the presentation as well to talk more about that. But it is basically, I work day and night, it feels like sometimes, starting these out from just a liquid solution to something that you could, even though it's so small, you can physically hold it. And so it's really cool being able to work alongside someone who, seeing them reach their goals of cardiovascular health and being able to have it be this whole, founding thing for cardiovascular health is super, super cool. So this is my fun area of research. So technically, I work in the Division of Developmental and Stem Cell Biology. So again, that is mainly in the IQ building. So like I've said, we focus mainly to improve cardiovascular health tissues. So basically what I focus more on is neurocrest cells. So when you have a human that is forming, you have roughly, you can say, like your spinal cord, and you have a tube within that, and then you have neurocrest cells that can stem out from that go to your different organs and different parts of your body. And what I'm focusing at is more of, preventative rather than like treatment, which is like really cool. So when those certain cells go onto your heart, a lot of congenital heart diseases can be from basically that path gets obstructed when they are going from, being created to their destined organ. That when that gets disrupted because of certain things, that is what causes a lot of congenital heart diseases. So by being able to make the mini models of the heart, and then we can inject them with the neurocrest cells, by doing that, we are able to model what that looks like in humans without the need of, without the needs of just like having it be like an online model, or, if someone were to choose to donate their organs. It's very, very fundamental being able to see that in the development phase, which is what we are able to do. And then this image is just like a staining of like a tissue that we use like different fluorescent dyes. So we can do subsections, we can cut the mini organoids, and then we can look at their tissues underneath the microscope to see if the neurocrest cells have reached their destinations without any issues. And then you can see that here in the green, which is just stained with GFP, which is, I'll never escape GFP. It's part of my life forever probably now. And then this is my fun lab. The one, I don't know what size of the picture that is for you guys, maybe the right, but the one standing up behind the person in the black coat, that's Sasha. That's the person that I trained with, that I have been with since my sophomore year. So what I'm looking specifically at now, I actually just submitted my third paper, is about how an imbalance in lipids can cause a bunch of cardiomyopathies, which is basically just it can be a group of cardiac health diseases where you have especially like enlargement of the chambers, like the bottom ones of your heart, and just, a broad range. And so we're currently waiting for the review back for that. So these past few months I have just been doing like housekeeping experiments, just keeping things, growing the organoids, keeping them, in stable conditions as much as I possibly can. So we're hoping to get that review back very quickly, hopefully. It's been a few months, so fingers crossed. So that was really interesting being able to look at, because that was a more project that she let me take more hands-on about. I was allowed to take the lead in a lot more of it, which was really, really exciting to kind of, even though being like an undergrad, like I still get to do cool things, but I almost got a taste of the PhD role for a second. And I was like, ooh, this is cool, guys. Like, whoa. And then this slide is my favorite. This is like the why it matters. This was like why I, chose to do research. So I actually struggle with cardiovascular health conditions myself. I have SVT, which basically means my ventricles don't like me. They don't like to work super fast. My heartbeat cannot regulate itself. And so being able to, work alongside all these people and like being able to see the impact that comes up it's really inspiring to know that like there's hope in the future, that people won't have to go through what I've had to go through growing up, or different preventative or treatment methods. So that's really, really exciting, and it means a lot to me being able to I don't know, just like being able to see it. It's like it's hard to put into words, kind of like experiencing something that can be really difficult to manage, and then knowing that you're being a part of the group of people that are making a positive impact in the change for it. The long-term goal is to be able to create fully functional adult models of the heart. Right now, I say that is like science fiction, because just the reality of it is we don't have the technology available to us yet in order to have the heart models get that large. But I do thoroughly believe that it will be fully possible in the future, and the goal of that is maybe even possible for transplant in the future, which would be amazing. So if you're on like the transplant list, you don't have to wait for like so many people need transplant organs, and it would be amazing to be able to have one fully functional, ready to go whenever you needed it, rather than that feeling of uncertainty of being on a list waiting to see if someone has one for you. And then also just kind of what we're doing now is preventative medicine, so therapeutic approaches. If we have a model, then we can just grow it, and then we have one there at our disposal to just be able to look at a little more in depth. And again, being able to have it in such a quantified amount would be very, very helpful. when this becomes a reality, it will be able to help millions of people. People like me too, where it's like, dealing with, it might seem like a small cardiac issue compared to so many others, but being able to help small and big issues is a really, really good and really important. So why I chose this research, like I mentioned before, was because I struggle with cardiac issues myself. But I remember, I'm sure my fellow students know the struggle of going on Handshake, trying to find research positions. It's a struggle. And so I remember I saw this one and it immediately popped out to me, and I was like, oh, if I could get this, that would be phenomenal. And I remember I emailed the PI that was for it, and I emailed him, and like 5 minutes later, he emailed me back. He was like, I don't need your resume. Do you want an interview? And I was like, oh, yeah. I was like, okay, cool. And I showed up like with my resume, and I was like, here. And he was like, no. Well, he was like, so why are you here? And I'm like, okay, thank you. Really intimidating, but he's a great guy. And so he gave me it like a week later. He emailed me back, and I was so excited. I was in the Minskoff studying, and I fell on the floor. I was really excited. I remember I was actually studying the cardiovascular unit in a class. So I was like, whoa, universal lined up. But it was super, super cool. And then after graduation, so my mind actually has probably changed since when I made this presentation. I feel like every time I wake up, my mind changes. But I graduate this spring, and I know for sure I'm at least taking a gap year. So excited to not have to have exams. But speaking of, I probably will take the MCAT. Maybe. I'm figuring it out. I wanted to go to medical school ever since I was a kid. I knew I wanted to do something medical, but then I started my research, and then I was like, oh, actually a PhD sounds like it would be even better. Super fun. I know they have MD, PhD programs. If you know anyone who's did that, give them a hug. They're in school like 12 years. It's a lot. But I know that for sure it will be something in cardiology, because that's just a very important field to me. And I will do research no matter which path I end up taking. So we will see. But thank you. And then if anyone has any questions.
Thank you, Haley. There were a couple of online questions that came through. How long can you keep a stem cell heart alive?
That's a good question. So I like to say that they are like newborn babies. I call it bath water. They have this media that they sit in, and I have to change it at the exact same time every single day. Or else if it gets too old, then it's kind of like, if you have produce and it sits in like its juices for too long, it can get bad. It's literally the same concept. That was how my PI described it to me, which is a great way to memorize it. So it completely depends. Usually it takes them 40 to 48 days to grow, so they can stay, functioning within that time frame for sure. And then after that, we usually give ourselves about a week to work with them, because since they're so small, they can't, function for a very, very long time before they just eventually just stop working. So by the time they get to around day 40 to 48, we have a week to do our imaging, our tests, all of that stuff. And then after that, it was when they would stop functioning. So like the image I showed on the first slide, to get those pristine of images, we would probably do that right on the first day that they were done being fully developed. And once they stopped growing, and then we would take their images, and then within 5 to 7 days they're
Does anyone else have a question before I ask one more? Okay. Just have time.
So I have a quick question for you. I'm just absolutely fascinated, this idea about making these sort of, mini hearts, right? It looks like the bottom of 96 well plates, right? You got a little tiny one down there. And so is it just like you start with a single stem cell and you let it grow? Just if you go into a little bit more detail on the process of that.
So by the time that I'm allowed to work with them, all of the stem cell part of it, I don't know what they're specifically called, but instead of a well plate, they have flat bottoms, and they're kind of in like a tray looking thing. And my mentor is actually the one that does that, because it's a very expensive part, and she trusts me, but I almost don't trust me because of how expensive it is. So by the time that I get it, at the bottom of like the flat surface, there's like a layer of cells, kind of, that you can't see with your naked eye, but like the media color is a little bit different that you just get trained to see. And so you take off the liquid, and then if you kind of use like they're like suction tweezers almost. And then if you grab them and then if you put them in the well plate, kind of, you can drop them into the media because they're also still very, very small. And by the time that they're in there, we have this pink liquid. It's really nice having it colored. I never would appreciate colored liquids until I started working in this lab because you can actually see. And then they kind of just, like the well plate picture that I showed you, that's where they stay. And we have an incubator that they go in as well. And they kind of just sit there on the bottom, and then we just change the media again every day. And that also helps guarantee that when they're growing, they don't grow and stick to the bottom of the well plate, because then you can't. They're stuck there, unfortunately. So the media is infused with kind of like different proteins and stuff. So when we do different medias for different stages, and so when the media goes in there, it kind of like absorbs it a little bit, and almost like it doesn't like secrete the unwanted ones, but it kind of like lightly leaks out a little bit. And so that's why it's important we take the media out, and then we let them sit there, and then they grow, and then they eventually reach that size. They're a little bit bigger now than from that picture, which is really cool. I've been able to see them grow throughout my time. And then they just sit there in the liquid. And then when we transport them onto slides for imaging, when we use pipette tips, but the organoid can't touch the plastic part or else it falls apart because they're very, very fragile. So that's the hardest thing I've ever done in my entire life. But basically they sit in fancy water and grow is how I explain it to most people. It's like those washcloths that you buy at the Dollar Tree that are hard, and then you put them in water and then they expand and then you can use them. It's like that, but with hearts. Super cool.
All righty. Thank you, Haley.